Lipids are a type of biomolecule. Some examples of lipids include fats, oils, phospholipids, steroids, and waxes. In general, the function of lipids in living organisms is as a long-term storage for energy and carbon. Lipids contain the elements carbon, hydrogen, and oxygen. But unlike carbohydrates, there is no specific ratio of hydrogen to oxygen in lipid molecules. Typically, lipid molecules are insoluble in water. And the reason for that is they are nonpolar molecules, and we know that nonpolar molecules are not attracted to polar water molecules. And lipids are made up of building blocks called fatty acids and glycerol. So let's look at how a lipid molecule is formed. So over here on the right, we have an unsaturated fatty acid. And let's just break that down for a minute. Let's look at the term unsaturated. We've seen that term before when we went through our organic chemistry. Unsaturated means that we've got a double bond somewhere along the hydrocarbon chain. And we've got the COOH at the end, which makes it an organic acid. If we look at the two fatty acids down below, they're considered to be unsaturated fatty acids. And unsaturated fatty acids are ones that have single bonds throughout their hydrocarbon chain. And they both have that COOH at the end, so they're a type of organic acid. Now, fatty acids are bonded to glycerol molecules to make lipids. So if we look at the word glycerol, it's got that OL in it, and that should be a familiar suffix, which tells us this is a type of alcohol that contains hydroxyl groups. So what we're going to see when these glycerol molecules and fatty acid molecules bond together is a process called dehydration synthesis. We're going to lose an OH off of our acid and a hydrogen off of our glycerol. That hydrogen and OH is going to form a water molecule, and a bond is going to form between the glycerol and the fatty acid. The same thing would happen here. An OH would be lost, a hydrogen would be lost, they would come together to form a water molecule, and a bond would be formed. And lastly here, we'd lose an OH and an H, a third water molecule would be formed, and a bond would be formed between the fatty acid and the glycerol. So when we look at a lipid is made out of fatty acid molecules bonded to a glycerol. So a water molecule was lost to form this bond, a water molecule was lost to form this bond, and a water molecule was lost to form this bond. So it's a dehydration synthesis process. So when we look at saturated fatty acids, again, we're going to see single bonded carbons throughout the hydrocarbon chain. Saturated fatty acids tend to be solids at room temperature, and some examples of saturated fatty acids are butter and lard. Unsaturated fatty acids are going to have double bonds somewhere along their hydrocarbon chain. They tend to be liquids at room temperature. So oils are unsaturated fatty acids. Olive oil, corn oil, sunflower oil are all examples of unsaturated fatty acids. Now, phospholipids are another type of lipid molecule. So we have the word lipid here, but in addition, we're going to have phosphate groups associated with our lipid molecules. So phospholipids contain two fatty acids, a phosphate group, and that glycerol. Phospholipids tend to be the building blocks for cell membranes, so you'll talk a lot about them in your biology class. The phosphate group will be at one end of this phospholipid molecule, and because they are polar groups, they will be attracted to polar water molecules and will be soluble in water. But the fatty acid section of a phospholipid molecule, which is nonpolar, will be insoluble in water, or we'll refer to them as being hydrophobic, which kind of means they're afraid of water, or they're not going to be attracted to water molecules. So let's look at the general structure of a phospholipid. So up here along the top, we have a nitrogen-containing compound. Then we've got our phosphate group here in the middle. We've got our glycerol molecule attached to two fatty acids. And if we look over here, we've got another version of what the molecule would look like. So we've got the nitrogen group, the phosphate group, the glycerol, and the two fatty acids. This region up here, which is sometimes referred to as the head of the phospholipid molecule, is water soluble. Because it's polar, it's going to be attracted to those polar water molecules. This region down and along here, which contains the fatty acids, is insoluble in water or soluble in fats. Now, a shorthand way that this molecule is drawn in biology is something that looks like this. So this region here is sometimes referred to, as the head of the molecule, and these are referred to as the tails. The head region would be attracted to the water molecules, whereas the fatty acid or the tail regions would be repelled by the water molecules. So let's look at glyceride molecules. Glyceride molecules are energy sources for living organisms. They're also used as insulation against cold weather. They're physical protection for internal organs. And they're stored fat deposits, which our bodies must break down before they can be used. Prostaglandins are another type of lipid. They have fatty acids that are joined together in rings. And when we look at these prostaglandins, they are a coordinator between cells. They help to direct local cellular activities. They're associated with things like pain and labor contractions. Steroids are another type of lipid. And again, they're going to be ringed structures. Cholesterol is a good example of a steroid. And cholesterol is found in the structure of cell membranes, in hormones, but cholesterol molecules are not found in plant cells. Another example of a lipid are waxes. And when we look at waxes, we're going to be looking at lots of fatty acid chains that are linked together in long chain backbones. They're waterproofing for plants and for birds on their feathers. They produce honeycombs, and they're found in human ear canals.